So my name is Mark Swanson, and I'm a graduate student in Jacob Esselstyn's lab at the LSU Museum of Natural Sciences, and I'd like to emphasize the importance of the contributions of our colleagues at Museum Zoologicum Borgonese near Jakarta to the outcomes of this project. So it's becoming increasingly clear about that interactions, long-term interactions between mammals and their microbial communities are really important to mammalian evolution and fitness. And so some examples of this are that mammals produce in their milk particular sugars that can only be digested by bacteria. And so mammals are potentially feeding bacteria in a way that encourages a specific community in their offspring's gut. Additionally, polysaccharides are the most abundant, or cellulose is the most abundant polysaccharide in the environment. And so mammals need gut microbial communities in order to ferment those complex polysaccharides down into short-chain fatty acids that can be readily absorbed by the host for energy. And finally, there have recently been a lot of mechanistic studies showing that presence or absence of particular strains of the Bifidobacterium have important consequences for proper development of the immune system and behavior in rodents. So despite the importance of all microbial communities to mammalian ecology and evolution, they've actually been looked at relatively little in wild systems. And one of the reasons for that is because of how many different factors have impacts on microbial community composition, including host species, the host diet, geography, host condition. So a lot of researchers are probably concerned that without a properly designed study that there's just going to be noise, or it's hard to pull any information out of the data that you get. And because of this, some very basic questions about microbial communities in mammals haven't been looked at in natural populations or in a comparative context. So we know that there's concentration gradients across the length of the gastrointestinal tract in pH and oxygen concentration, as well as the presence of antimicrobial peptides. The vast majority of sampling for mammal microbial communities comes from fecal samples in the colon, but it's possible that because of that really high concentration of antimicrobial peptides, that the host is more carefully controlling what lives in the small intestine than in other regions of the gut. And so we could see the strongest signal of host taxonomy and phylogeny somewhere else in the gut that's not from poop or feces. So in order to get at this, we wanted to ask the question, which region of the gastrointestinal tract shows the most taxonomic signal, as well as asking whether microbial community abundances show any phylogenetic signal at all. And we're also interested then in which microbial taxa are related to diet when we take the phylogenetic structure into account in the analyses. So the tropics are great places to look at questions like this because they have very species-rich communities, a lot of species that are related at different phylogenetic scales and have a diversity of dietary strategies. And then you can sample all of those species from one location without having geography as a confounding variable. So for this project, we sampled 8 species of rodents, 1 squirrel and 7 murines from a mountain on the northeastern peninsula of Sulawesi in Indonesia. And so next I'm going to start showing you some pictures of the animals that we sampled that make up this community. So this is Procyrillus murinus. This is a squirrel. And the only records we have of its stomach contents are moth larvae and fruit. This is Echinothrix leucura, which is a worm-eating specialist, thought to only eat worms and insects. Rattus antherus is thought to be a frugivore, and both Parodomes dominator and Pteromes tyrae are also thought to be frugivores or eat primarily plant material. Bunonis fractorum is thought to be an omnivore, eating primarily, or eating any insects and plant material available in the environment. And both of these Maximus species are also thought to be omnivorous, eating whatever plant or insect material is available. All of these, all this dietary information is from only one person's observations during a few years at one site in Sulawesi. So we do need to collect additional dietary data to confirm if those observations are accurate in order to do this work. So we looked at the diet of these animals by getting stable isotope data from the hair, specifically nitrogen-15. In order to sample the microbial communities, we swabbed the contents and host intestinal tissue from the small intestine, cecum, and colon of 2 to 7 individuals per rodent genus. We identified the bacterial taxa by sequencing the E4 region of the 16S RNA gene in accordance with Earth Microbiome Project protocols, and the sequences were clustered into 97% OTUs. So this is our stable isotope data, and so higher levels of nitrogen-15 mean that the animal is feeding at a higher trophic level relative to other members of the community. And for Echinothrix lucura, that worm-eating specialist, in yellow, we see that our expectations are accurate, that it is feeding at a higher trophic level relative to other members of the community. What's surprising is Bunomius fraterum in red, which we thought was an omnivore, is actually also seems to be feeding on primarily insect material. So that was a surprise and a good reason that we collected these data in order to confirm that. All of these with lower levels of nitrogen-15 are the putative herbivores and frugivores that we talked about. And interestingly, Procyrillus murinus, which there's only records of it eating moth larvae and fruit, falls out right in between the insectivores and herbivores. So it seems like this matches up pretty well with our expectations. So this is an ordination plot of the Bray-Curtis distances between 16S RNA samples, and this is from the small intestine. So we see some degree of clustering. So these are all the Maxine species in green and the Bunomius species in red, but kind of relatively weak associations by host genus. We can quantify the degree to which these samples group by host genus by calculating an anosim R statistic. So in the analysis of similarity, where a value of 1 means that the samples are highly distinguishable by host genus, and a value of 0 means that they're randomly distributed across the plot. So for the small intestine specifically, we get an intermediate value of 0.52. So there's some degree of clustering, but it's relatively weak. What we were pretty surprised by, and was really cool, was seeing really strong clustering of microbial communities by host genus in the cecum particularly. These are all the worm-eating specialists, Echinothrix lucura. And so we see all of the, not only are they grouping by host genus, but we also see all of the insectivores on one side of the plot and herbivores on the other side of the plot. And the degree to which these are clustering by host genus is reflected in this anosim R statistic of 0.92, which is pretty close to 1. And in the colon then, similar to the small intestine, we see some degree of clustering, but it's still relatively weak, and an anosim value of 0.56. So we can put this into context by looking at how these microbial communities cluster by host taxonomy across the length of the gut. And the only previous work that's been done like this in a comparative context was by Kevin Cole in his 2018 molecular ecology paper. So what he did for this project was look at 7 different species of Paramyscus, which were all raised in a lab on a controlled diet in order to not have that as a confounding variable. And he found really high anosim values, degrees of distinguishability all across the length of the gastrointestinal tract. So it's pretty exciting that even though the animals we were sampling were from natural populations, they weren't on a controlled diet, that we still see pretty high degrees of distinguishability by host taxon in the cecum. So just because people working in natural systems might be sampling from the fecal feces and colon tissue and not seeing a relationship to host taxonomy or host phylogeny. It doesn't mean that signal is not there. It might just exist in a different part of the gut. In the cecum, we also see much higher levels of community evenness and community richness. So it's possible the reason we see this stronger signal is because the communities are more diverse and more stable, less likely to be perturbed by environmental influences, changes in nutritional availability, and host condition. So the fact that we see all of the insectivorous taxa on one side of the plot and the herbivorous taxa on the other side of the plot might make us want to ask which microbial taxa are associated with insectivory or herbivory. And so in microbial ecology, this is typically done through differential abundance testing implemented in DC2 or through network analysis in order to infer associations between the abundances of particular microbial taxa and diet. But first, because these are all coming from species that are related to each other at different levels, we need to ask whether there's phylogenetic signal present in the relative abundances of these gut microbial communities. And if so, we would need to take into account methods which include phylogenetic structure. So I just want to emphasize that another Esselstyn lab member, John Nations, spearheaded the following computational work. So we use our package, BRMS, to do Bayesian multi-level modeling, which allows us to look at the effect of the nitrogen-15 or trophic level on the abundances of particular microbial families, given both the intraspecific variation and the structure of the host phylogeny. And so we looked at this with the relative abundances of 7 of the most commonly encountered microbial families in our data set. And what we get here is a distribution of Hagel's lambda values. So Hagel's lambda is a measure of phylogenetic signal, where 1 indicates high phylogenetic signal, and that the trait is evolving along the phylogeny, whereas a value of 0 indicates low phylogenetic signal, and that traits are evolving primarily in response to environmental factors. And so for 3 of the families in our data set, we get a really narrow range of posterior Hagel's lambda values, which indicate that there's a lot of confidence in high phylogenetic signal. And then for another set of 3 families, we have less confidence in that high value, and one of our families, we have too much error to be able to say with confidence how much phylogenetic signal there is. So what's really interesting is that when we take phylogenetic signal into account, we regress the nitrogen-15 trophic level values against the relative abundances of microbial families. The only family where we see an association between abundance in the gut and herbivory or carnivory is in the Homothermaceae. And this is really interesting because this family is found almost exclusively within mammalian intestines and is known to degrade plant lichens as well as host-produced mucins. So I don't know where I am for time, but okay. So with that, I'd like to thank you all for your time and emphasize the contributions of my lab mates to both the computational work and the sampling. I'm not sure if I have any time for questions.
Nope.
I'd be happy to take them.